our next speaker needs no introduction because you know him well. But for those who haven't been at JGI very long and those aren't part of JGI, I'll give a quick introduction to Dan Drell. And back on Monday, I introduced Ari Petrinos as our founding father of the JGI. And in a similar vein, Dan Drell is our sagely guardian uncle. Okay. Dan served as our program manager for 13 years from 2005, was a great partner to us, friend to many of us, and continues to be a strong advocate for us. And so Dan has a bachelor's in biology from Harvard, PhD in immunology from the University of Alberta in immunology and genetics. And he spent 27 years at DOE before retiring in 2018. And I'll just share a little personal thing here, because one of my regrets in taking this job is at the time I didn't have that long to work with Dan. And we have both a shared love of science and perhaps more importantly though, a shared love of a certain distilled beverage from Scotland. And if we worked longer together, who knows what we would have come up with. And so, Dan, please.
Well, thank you very much. It's a real pleasure to be here again, 3 years and one pandemic after I attended as a fresh retiree the 23rd annual meeting. I want to begin with thanks, of course, to Nigel for the invitation to be here. There's still a lot of malt scotch out there, and you will never lack for someone to enjoy it with. Who could have imagined what's happened since that last meeting, pandemic and lots of other events. But before going any further, I want to make sure I acknowledge and thank Denise Yaden, Wendy Lynch, and Marilyn Huey for organizing and supporting under very difficult circumstances this tremendous meeting. My role during the Human Genome Project was to manage a small but important program element. How about that? I mixed a page. So you heard on Monday from Ari, who oversaw DOE's contributions to the Human Genome Program during its most critical and difficult years, and deserves, in my view, most of the credit for DOE's role and for the détente between the government program and the Craig Venter independent effort that permitted President Clinton in June of 2000 to announce the completion of the first draft. The president announced it with Francis Collins and Craig Venter at his side. I've always thought Ari should have been up there with him. It's frightening now to think, imagine if you will, how 2000 and the Genome Project might have ended were it not for his heroic efforts. It wouldn't have looked the same. So my role during the HGP was to manage a small but important program element looking at ethical, legal, and social affairs. And as the Human Genome Program wound down, I transitioned into microbial genome programs, and that's where my association with JGI began. We can debate how much impact LC programs, as they were called, had, but they were good at raising some of the important issues that came with increasing skill and prowess at sequencing human genomes and elucidating some of the personal identifying information that is in each of our genomes that has medical and wider implications. Many of these issues remain with us and have been exacerbated by continuing advances in biotechnology. And I want to give a special shout out to Jennifer Doudna, who we heard from on Monday, for her leadership role in looking at these issues with respect to CRISPR. It's important work and discussions about its meaning for all of us should not be limited just to the experts. The implications affect all of us. So as Nigel mentioned, I was the JGI's program manager from 2006, I think, to 2018. And while I never said this out loud, especially in BER, I think I had the easiest job in the office. all, and I do mean all, the credit for that goes to the terrific people here at the JGI: Eddie Rubin, Jim Bristow, Ray Turner, Axel Vissel, Susanna Tringe, Len Pinocchio, Nicholas Kirpides, Kirsten Fagnan, many, many more, who collectively put fewer demands on me than any other user facility program manager. It's easy to look good when you have great people doing a great job anticipating the challenges and addressing the problems early when they're small, easier to deal with, and before they grow into big headaches. Not only this was true of the science that the JGI did, but under the superb leadership of Ray Turner, Joe Harkins, and many others, the integrative genomics building was planned and built on time and under budget. And I need to give credit as well to my good friend and former colleague, John Houghton, who oversaw it initially at headquarters, except for that missing walkway. And Nigel and his team have continued that excellence. I'd be remiss if I didn't also call out for special appreciation the project management office. Krista Pinocchio, Tootie Tatum, Carrie Berry, and all the wonderful project managers made my job back in Washington so much easier because of the calls I did not get. I'm referring to the JGI isn't helpful. I can't get my work done. They're hard to work with. I got lots of calls from scientists whose projects didn't get accepted. I expected that we could deal with. But I got none about the processes of actually working here. At meetings where I encountered JGI users, I would routinely hear praises for how wonderful the PMO was and how hard they worked to help make a user's project achieve its goals. That to me was a huge and ongoing success measure, not an easy one to measure or quantify, but of surpassing importance. And I will say that if there were a PMO Olympics, JGI's team would win the gold medal every time. For BER and the communities of scientists that you serve and enable, though, it's really the science. With very rare exceptions, scientific impact takes time. And to appreciate it and understand it doesn't happen overnight. Now, note I said scientific impact. I did not say scientific measures. And that's because arguments on how best, if at all, to assess or evaluate the value of any given scientific effort quantitatively have been around since the ancient Greeks, continuing to this day, and they're going to be going on long after we're all gone. Much as politicians or bureaucrats, and I'm pleased to say I'm neither anymore, functions and funders, public and private, want quantitative and predictive measures. I don't see any reliable ones anytime soon, which is why they like numbers. And after the fact, anecdotes, though informative, can be hard to generalize in specific actionable ways that can appeal to a legislator. The best anecdotes to me have always seemed to have the element of wonder. There are so many examples, I'm going to pick two. So when Hamilton Smith in his lab at Hopkins in the late 60s saw ladders of DNA fragments on his gels instead of a smear, he said, or words to this effect, "Gee, that's strange. I wonder what's going on." The consequence of that was the discovery of the first restriction enzymes, and the revolution in biology that's enabled is still with us, and it's changed our world. When Jill Banfield, among others, saw these strange repeated segments of DNA in bacterial genomes, many of which were sequenced right here, called clustered regularly interspersed short palindromic repeats. I will say as an aside, whoever came up with that term wasn't a poet. And then met up with Jennifer Doudna for that now famous cup of coffee at the Free Speech Movement Cafe, not that far from here. Again, the spark of inspiration resulted in a transformative change in our world. And I would characterize it as a near thermonuclear biological revolution. And to echo a point I made a few minutes ago, along with extraordinary scientific advances, both of these revolutions have presented society and us with large challenges of what constraints, if any, we collectively devise to ensure that we see the benefits of these discoveries and not costs. So what these stories and many others like them all share is complete unpredictability. You can't plan for specific discoveries. You can't budget for a certain number, for a certain amount of money, much as lots of people back in Washington wish you could. And fundamental science is irreducibly unpredictable, and that's its glory. And that's why it can be so much fun to do. And sadly, why politicians and bureaucrats regard it with such frustration and sometimes distrust. So I have one overhead to show, with your help, Wendy. It is, thank you. Now this overhead's gone through a couple of iterations since Monday. Thanks first to Nigel and then to Chris Daum yesterday at the tour. But the upshot since 1997 is that JGI's productivity has increased 30 million fold. And what took JGI a full year to do in 1997 now takes JGI less than a second. And that is slated to increase. We took great pride in that 20 million base pairs back then too. It was important both to show that DOE was a serious participant in the Human Genome Project and that progress towards the goal of a complete reference sequence was being achieved. But there's some cautions here. Though sequencing productivity became the primary measure of achievement, we needed to be careful because the Human Genome Project, I've analogized to the march down the field in a football game. You know where the end zone is, it's 100 yards away, 80 yards if you touch back. And yardage is an obvious, ready, easily agreed on measure for progress moving down the field. It's appropriate for a project that wasn't fundamental research because there was never any doubt there was a genome. We knew what it was made of. We knew roughly how big it was. We didn't know how many genes there were, and there were lots of fun discussions of that number. And while there was a lot of research necessary to build the tools to achieve it, and then interpret it and compare it to genomes and fragments that we already knew about, there wasn't any doubt that it was conceptually achievable. It was a construction project. It wasn't like looking for a subatomic particle that theory predicted might be at a certain energy, but you still didn't know and might not exist. So we were building a tool. Given that the accepted measure of genomic achievement was sequence production, however, the numbers in this graphic are astonishing, and they became the coin of the realm. Again, just in an earlier calculation, I looked it up. There are 31 million seconds in a year. 600 million base pairs means less than a second. It's astonishing. It's really astonishing, but it also presents a challenge, because what do you do with that capability? One thing that was exciting about the early days of microbial sequencing was the unexpected findings. And I'll always remember a meeting many years ago when Derek Lovely reported on the sequencing of Geobacter sulfuriducens, and he put up a slide that had unexpected revelations from the sequence. This was 20 years ago, but it was typified by a complete inability to identify genes for microbial functions that the bugs displayed every day in tissue culture. It was also that he found numerous genes for capabilities the bug never used, never showed. And this was just the beginning. Myriad genomes since then have revealed similar and even more surprising things. The genes for these undisplayed functions were there, of course, but so different in sequence and evolutionary origin that they just couldn't be recognized. But the true evaluation, and again, I avoid the word measure, of the JGI is the science that this amazing sequencing rate makes possible. And the last 2.5 days have illustrated that better than I can. Keep in mind, it isn't just the quantity of sequence you generate, it's where you go with it. And that's where JGI's record's been extraordinary. And that's why it was so much fun for 13 years to be associated as program manager with JGI. I had initially thought of listing a few examples. I'm not going to do that. I'm going to take my example from Todd Anderson's talk yesterday and simply throw up my hands and say there are too many to select. And that's an astonishing statement and one to be very proud of. So it isn't just the generation of vast troves of data, it's the new areas of scientific possibility that these accomplishments open up. The extraordinary JGI throughput has also made possible super programs in uncultured microbes, diverse plants, fungi, algae, my personal view most powerfully, the diverse metagenomic initiatives that we've heard a lot about. I think the FICUS initiative is particularly promising, and it was very encouraging to me to hear Todd talk about it so positively. And I thrill to the number of talks in the last 2.5 days that have had some mention of it. The variety of new technologies that JGI is bringing on board, past and present, is extraordinary and amplifies what these programs can do. So in the last 25 years, JGI has changed biology. The reach of your science is apparent in the examples that have been displayed at this and past user meetings. Importantly, your reach is far from over. It will last a very long time because the nature of your work is foundational in the best sense of the word, exploratory. It may be trite to say this, and I hope Paramount won't come after me, but what JGI has done for so many years is to, quote, boldly go where no one has gone before. And quite frankly, that's the best characterization of your science that I can come up with. I'm encouraged to learn you're initiating a strategic planning effort. I think this is important. It has to happen at periodic intervals, and it's about time for it to happen again. And sorry, Nigel, but uninvited, I'm going to offer a few thoughts. One is to consider what we always called the Eddie Rubin deletion test. Namely, what wouldn't happen if JGI were to stop functioning? After answering that question, you prioritize what's most important, focus on addressing that. Another is that sequencing continues to be more ubiquitous. So what is it that only a JGI-sized place can do? You don't need a JGI to sequence a single microbe or even a small number of microbes anymore. You need a JGI to sequence something that takes trillions of base pairs to answer your scientific question. What are those questions? What could you do if FICUS or a program like it were given a shot of steroids? There's not much point in reinventing wheels that other user facilities or labs already have. And so building more connections with other user facilities is simply efficient, easier and cheaper. As several speakers have shown, there's a vast undiscovered diversity of genes and therefore gene products. And therefore metabolites, enzymes, pathways, et cetera, that can address many of the problems we already have So who needs to reinvent the wheel again if the, sorry to X-Files fans, if the truth is out there, so to speak, buried in dirt soils elsewhere, it may be cost effective just to keep looking for it. Think big. I mean, this was said earlier this morning, but it's true. And in terms of JGI future directions, hopefully this will resonate with at least one member of the audience that you can borrow from Wayne Gretzky and plan on skating to where the puck is going to be, what's out there in the future. But another way of saying, perhaps a little more literate, is from George Bernard Shaw, who said, "Some see things as they are and ask why. I dream things that never were and ask why not." I think it's JGI's challenge and role to ask why not. It's been a privilege. It was a privilege to work with you for the 13 years up to my retirement. I know Ramana Madupu is continuing to look after you well, though I must say I'm in awe of the huge challenge she and all of you have had to deal with COVID. Your first 25 years have been terrific. I'm confident your next 25 years will be as good and even better. And thank you very much.
Thank you, Uncle Dan, for everything. Much appreciated.